All right, hello everybody and welcome to our third session in the Modeling Biology Domain Seminar by the Center for Reproducible Biomedical Modeling. Today we're going to be covering the domain of cardiac tissue. I'll just go through some of the logistics of the seminar and a quick introduction on the domain. So if you've attended some of our previous sessions, you know that we are trying to break down several biological domains so that we can get opinions from multiple experts on that domain to understand how the field is being modeled. We've gone through 2 sessions so far that covered protein signaling and gene expression, and now we're moving into domains that incorporate multiple scales of biology, and cardiac tissue is a really great example of that, as you'll hear from our speakers. Please join us for upcoming sessions. We have several coming up that may interest you depending on what domain you're working in. So our seminar format, we're going to go through a quick introduction of the topic. We'll have 2 20-minute talks from each of our speakers, and then we'll conclude with a group discussion. Our general goals for our seminar series are to identify common challenges in the domain of cardiac modeling, share our personal experiences and solutions, brainstorm new solutions, and then, if we can, create a collaborative community of modelers. So hopefully, if you're working in cardiac tissue modeling, that you'll meet some other modelers here. And then guidelines, always err on the side of commenting. We really want to hear your opinions on the talks you're hearing and your questions on the topic. Honest debate is encouraged, and please mute your microphone when you're not talking, just so that we can hear from everybody. All right, so today's domain is cardiac tissue. Modeling cardiac tissue is quite complex because it incorporates multiple scales of biology and many mechanistic, biophysical, mathematical descriptions in order to fully understand the system that we're working with. Here this is an example of how you might generate a patient-specific model, but whether it's patient-specific or not, you're going to be incorporating data that comes from multiple data sources. It could be whole organ imaging through MRI, it could be blood pressure measurements, genetic screens, and then you'll be incorporating information about the physiology and the physics of the system. So if you're incorporating genetic information, you might have some idea about ion channel mutations in your system. And then for the physical information, you're going to be looking at how ion flux is occurring through channels and information about the pressure through the cardiovascular system within whole organ models. And then hopefully once you have all this information, you'll be able to generate some ideas about the prognosis for individual patients or general ideas about how the cardiac tissue performs as a system. So a challenge with cardiac tissue modeling is the multi-scale biological nature that we talked about, and then also for different patients and within the tissue itself, heterogeneity in the tissue structure. So to illustrate the idea of this complexity, we have here a subcellular representation, and you can see that even in just cardiac cells, you're representing multiple spatial locations, and then also you're representing these ion fluxes. In addition to that, you can also explicitly model different mutations to ion channels, which is shown here in B, and you may also want to represent how a drug is influencing the system. So that is done here, and showing the dose response curve of how the current through sodium channels are affected by this inhibitor. Cardiac tissue modeling also has great opportunities. So we talked a little bit about patient-specific models. Depending on the data you're able to collect, we can hopefully use generalized models of the systems and build upon those if we have good confidence in them, incorporating patient data, for example, how their whole organ is represented, and then genetic information if we have that available, to then be able to predict how patients may respond to different treatments or drugs, for example. So today, to talk about some of these topics, we're going to have 2 speakers that are experts in cardiac tissue modeling. We're first going to hear from Dr. Miguel Aon, and we will then hear from Dr. Patrick Boyle. So let's first hear from Miguel, who is a scientist at the National Institutes of Health, to discuss his research in cardiac tissue modeling. Welcome, Dr. Aon.
Thank you.
One moment.
Can I share screen now?
Yes, yes.
Okay. You seeing the screen now?
Yes.
Okay.
We're
Hear me well?
seeing the presenter view, Miguel.
Okay.
Yeah.
So
Okay.
I think you probably need to toggle something.
Yes. Let me see.
Mm.
Oh. So let me see. But right now?
Yeah. No, almost. There was I think that you have to change something in the display settings to select the.
Um,
Now it's okay.
now?
Miguel. Yeah.
Now it's okay? Okay.
Yeah.
So, hello, everybody, and thank you very much for the kind invitation. And the talk of today, experimenting and modeling complexity in the heart at the interface of biology and physics, was originated and performed by this team of people at Johns Hopkins University and is continuing at the National Institute on Aging from the NIH. So one of the main underlying topic of this talk is the experimental computational synergy in which experimental results are simulated by a model for mechanistic and validation and further improvement. In turn, the model predicts new behaviors that can be corroborated by experiments. And several cycles can happen through this cycle as it happened in this work. So what you need to know, the organ address by this talk is the heart, of which the basic functional unit is the cardiomyocyte, an image of which is shown here, and the yellow spots correspond to mitochondria that you can see are arranged in a regular lattice across the cell. In 3D, a cardiomyocyte is like a brick of about 100 micrometers length, 20 micrometer width, and 20 micrometers depth, full of mitochondria, 15 to 20,000 of them. So it performs the basic function of the heart. It contracts, it relaxes, and contraction is when the pumps, the blood is, the heart pumps the blood through the distribution and circulation system, and relaxation is when the heart refills with blood and gets ready for the next contraction. So there are multiple scales of space in heart function. We have the whole heart, the tissue, the cell, and the mitochondrion, and also all these scales are somehow interconnected. There are also different time scales going from a few milliseconds to tens of seconds, and processes of a different nature like electrical, which is given essentially by a membrane potential measuring voltage, and they are faster than the mechanical ones, contraction and relaxation, and then even faster than the energetic ones, of which mitochondria function is supplying the energy for heart function. The scheme represents a basic view of the heart cell, in which we see the plasmalemma that in skeletal cardiac muscle is called sarcolemma, and we also see the contractile machinery and the power grid of the heart, which is the mitochondrial network. So it is important to remember for the next of the talk, for the rest of the talk, that these two membranes, the plasmalemma and the cardiac and the mitochondrial membrane, they are both sustained by different type of mechanisms, and so they are constantly changing, and we can monitor them separately. So the experiment that will be shown in this video is a key one because represents an important event for the triggering of arrhythmias. The elucidation of the mechanism involved and the scale up to the whole heart is what, and the computational modeling associated with it is what this talk is about. So we call the phenomenon that you are about to see in this video the mitochondrial criticality. And we see here a cardiomyocyte in which we will monitor simultaneously 3 signals, 2 corresponding to fluorescent sensors loading in the cells, and the third one, NADH, is an endogenous chemical from the cell. So the bright yellow spots that you see here are mitochondria, and when they are yellow means that they are they have high membrane potential, which is needed for effective function, and then we will have the ROS, the reactive oxygen species, which corresponds to reactive chemicals from oxygen chemistry. Many of them are free radicals, and the experiment occurs. We do a flash and then we will see in that region in which the mitochondria become blue, meaning that they are dysfunctional and they have low membrane potential. You can also see that there is an increase in ROS, and that a cluster of mitochondria seen here in green starts to develop. And then as the experiment goes, nothing seems to happen, but from the point of view of the cluster, you see that there is an increase and it starts to expand the whole cell. So at a certain point, then there will be a whole, there will be a whole blackout of the cell, and a new phenomenon will emerge there, which are sustained oscillations and synchronized throughout the mitochondrial network that happens at that critical stage. So from there, then this phenomenon can escalate to the whole network, and that starts in the mitochondria, goes to the mitochondrial network, and then can reach the whole heart and escalate to the whole heart, producing arrhythmias. And this mitochondrial criticality happens under ischemia reperfusion. And this scheme that I'm showing here has been proven experimentally and by computational modeling. And there is this escalation of failures have been shown by physicists to happen in systems at critical states like rockets, markets, and also heart cells, and in which those systems can crash, rupture, or I have shown completely collapse. Because in the case of the heart is a matter of life or death, we need to understand the mechanistic details of the phenomenon to be able to intervene. And these mechanisms will be assessed by the experimental computational synergy. So the mechanism of propagation of the mitochondrial loss of the membrane potential is called ROS-induced ROS release, and mitochondrial release ROS, and here is the superoxide anion, and which will increase the opening probability of neighboring mitochondria, and that therefore favoring the autocatalytic propagation. So percolation theory predicts that diffusion will become paramount locally between mitochondria that are distance only 1-2 micrometers between them, and then will propagate throughout the spanning cluster of the cell. Along with experiments, we were also modeling mitochondrial energetics and metabolism. And in these two papers shown here, we first described the basic bioenergetic processes of the mitochondria and tested the model for its capacity to reproduce fundamental energetic and bioenergetic behaviors. And the second model included the antioxidant systems and also the generation of ROS and the consumption by antioxidant systems. And then the exit of ROS through certain mitochondrial membrane channels. And it was able to simulate the oscillations as shown in the experiments. Then using this basic mitochondrial model, we arranged a lattice of 10 by 50, so a rectangular one of 500 mitochondria, and they were coupled by diffusion, in this case by superoxide, and we were able, and we asked if we were able to simulate the propagation of the mitochondrial loss of membrane potential. So for that, we increased the speed of acquisition of image acquisition, and we could resolve in time the depolarization wave as well as the repolarization wave, and as shown in the video. And then here is a montage of the depolarization and repolarization, and the model was able to simulate the 20 to 25 micrometers per second depolarization shown in the left of the membrane potential, of the mitochondrial membrane potential, and the increase in ROS shown in red on the right. So then mitochondrial criticality and oscillations can escalate from subcellular to the cellular level, And for testing this experimentally, we recorded simultaneously the mitochondrial membrane potential and the plasma membrane in a cardiac myocyte, which, as I said before, are independent and maintained by different mechanisms. So the graph on the left is a timeline image displaying the dynamics of fluorescence along a longitudinal line of the cardiomyocyte, where yellow means high membrane potential and blue is low membrane potential when mitochondria have lost the membrane potential. And so these blue bands going from one end to the opposite of the cell correspond to mitochondria that are synchronously depolarized, that is with low membrane potential. The yellow bands in between correspond to brief time of repolarization of mitochondria regain high membrane potential. And on the right, the plot shows the escalation of the oscillatory behavior from the subcellular to the cellular level, and which is visualized during the phase of mitochondrial loss of the membrane potential here in the blue bands, which is coincident with the action potential shortening in the recorded simultaneously, which you see here normal, and then in the blue band, when the mitochondria have lost the membrane potential, is dramatically shortened and becomes normal again when the mitochondria regain membrane potential. And this is shown by the in-phase relationship between the two variables. So we were ready then to integrate mitochondrial energetics to the electrical and mechanical processes in the cardiomyocyte. And this in silico version of a heart cell could reproduce contraction and relaxation, develop an action potential, simulate an increase in energy demand and supplied by mitochondria. In the first 2006 version of this computational model, comprising 50 ordinary differential equations, was able to simulate several important behaviors, both at a steady state and transient time-dependent states. It was further upgraded to be able to simulate the shortening of the action potential driven by mitochondria, loss of the membrane potential. And this version of the model was able to simulate the shortening of the action potential when mitochondria lose their membrane potential through the mechanism leading to the opening of plasma membrane channels, ATP-dependent potassium channels, or so-called sarcolemmal KATP channels. As shown here, during the loss of the membrane potential, the green line here, the action potential is drastically shortened, as shown here. So normal when the mitochondria are polarized, and dramatically shortened during the mitochondrial depolarization. So with this validated in silico model of the cardiomyocyte, we were now ready to scale up from cellular to tissue levels with the aim of simulating arrhythmias in the heart. We hypothesized that in the interheart, we'll find islands or clusters of cells in which their mitochondria have lost the membrane potential. And this cluster will create heterogeneity in the electrical propagation and presumably leading to arrhythmias. This image shows the intact cardiomyocytes arrangement in intact heart tissue called the myocardium, in which the cells are connected through gap junctions marked by the arrows here from tip to tip. And we were also able, by subjecting interhearts to ischemia, so the existence of big clusters of cells in which the mitochondria have lost their membrane potential. And so to test our hypothesis an experimental system was set up consisting of neonatal cardiac cells that proliferate and form cellular monolayers that have electrical activity, and therefore they are electrically coupled and able to conduct electrical propagation. Here, in these cardiomyocytes, the bright yellow spots are mitochondria. So those monolayers, as I said, are able to propagate, to perform electrical propagation, and as you see, is very fast. And then if we, in those monolayers, introduce a cluster of cells in which the mitochondria have lost their membrane potential, you will see that the electrical propagation comes, surrounds the cluster of cells that is the mitochondria depolarized, and they will be able at some point, here, to generate the spiral waves, which are the hallmark of arrhythmias in the heart. So then finally, I'll show you the scaling of the model of a cardiomyocyte into a two-dimension tissue model size of 5 × 5 centimeters square, having 63,000 nodes that of this model that I'm showing here, and then just in preparation for the video that I will show, and that will be the last one, is that we will simulate the existence of a cluster of cardiomyocytes that have lost their membrane potential, and then as a circle that you see here, and in which it is depolarized, and it will repolarize going into pink. And this is a much slower phenomenon because it's in a different time scale, whereas the electrical propagation is much faster. And so, and you will see then that as the electrical propagation comes, it will surround the cluster of cells, and then of a perturbation with the hallmark of arrhythmias. So this here is the main takeaway. So I hope to have convinced you that we should not avoid complexity, instead face it and work with it. It's fun. We have used an experimental computational approach, which is very powerful, and we have been rigorous and focused on mechanisms and verify them experimentally with the help of models that should be used not only to simulate, but to predict. And as you know, emergent phenomena cannot be predicted from the behavior of single components, but when you connect the function units with the right mechanisms, that can lead to successful multi-scale modeling. So to finalize, here are some links that I can provide where our models have been published or are in repository places. So, well, thank you very much.
Thank you, Dr. Aon. If you want to stop sharing, then we'll get Dr. Boyle's presentation going. I think we'll have a lot of good discussion later about the complexity.
Okay.
Yeah.
Stop. There we are.
Great. All right, Dr. Boyle.
Hi. Okay, so I first have to apologize because I'm a bit sick, and I live in Seattle. So because I have a cough, I'm sitting in my basement and not at my office. So I took a slightly different tack. Can everyone see my slides and my desktop and everything?
Yes.
Okay, so my approach is going to be slightly different. Rather than focusing on the applications of the type of modeling and simulation that we do in my lab here in Seattle, I'm going to focus on sort of the rudimentary concepts of the basics of modeling electrophysiology of the heart, usually human heart, but sometimes animal hearts as well. And I'm going to focus on the cell scale and tissue scale components of those models. And so the lecture part is going to be very high level. I'm not going to get down into the weeds, but I'd be happy to get into that in the discussion if anybody's interested. Or I can point you towards literature that can get you good and confused, like I am most days. And then the other half of the presentation, I'm going to be interleaving it with a live demo of the software that my lab uses. So the latest iteration of the software is called openCARP. It's a cardiac electrophysiology simulator that's freely available for academic purposes. And it's actually just had its first public release earlier this month. And so I figured it would be a timely presentation to this community. But CARP has been around for a long time in a non-open form, and it's what I've used since the very beginning of my PhD. So I can tell you a lot about how it works. So my lab is interested in studying cardiac arrhythmias. So on the left side of the screen here, I'm showing my favorite animated GIF from Wikipedia, which is the normal contracting heart. And what I want you to appreciate here is that excitation of the heart precedes contraction. So every single beat originates in the upper right atrium of the heart, which is where the sinoatrial node lives. That's the heart's natural intrinsic pacemaker. And spontaneous excitation initiates, propagates through the atria, aggregates at the atrioventricular node, which is roughly at the center of the heart, and enters the Purkinje fibers that line the ventricles, resulting in excitation and contraction of the ventricles. And the electrocardiogram, which is this ubiquitous signal traced out at the bottom of the slide, that highlights all the principal electrical events of a heartbeat. Atrial depolarization in the P wave, ventricular depolarization in the QRS complex, and ventricular repolarization and relaxation in the T wave. So if we lose this coordinated excitation, then the heart stops pumping, there's no oxygen delivered to the body, and you have sudden cardiac death within minutes. So arrhythmia, by definition, is any disruption of the heart's intrinsic rhythm regulation. Some arrhythmias are, of course, less lethal than others. Not every arrhythmia results in sudden cardiac death. And for example, atrial fibrillation is an arrhythmia that my lab has studied quite a bit. That poses no acute threat, but it's highly correlated with morbidity and other events, including embolic stroke. So what I want to stress here is that, and Miguel did an excellent job of introducing this, is that modeling electrophysiology is a complex multi-scale challenge. And so on the left here, I have the normal excitation of the rabbit ventricles in a computational model. This was from my PhD work. What I really want you to take away here is that the spatial scales and temporal scales that are relevant are on the order of centimeters and seconds, respectively. So the rabbit heart is about 2.5 centimeters from base to apex, and the time scale of a typical heartbeat, is on the order of 500 or 600 milliseconds. In contrast, at the cellular scale, a single cardiomyocyte is on the order of hundreds of microns long, or maybe even smaller, depending on the cell type. The cell type, and the temporal scales are extremely short. So the upstroke of the action potential takes place on the order of microseconds, and the events that happen at the subcellular scale, such as release of calcium from the sarcoplasmic reticulum, or buffering of calcium in different buffers, or indeed flow of different ionic species across the membranes of the mitochondria, those take place on the order of, with time constants on the order of microseconds. And so this can get pretty hairy pretty fast when we're trying to simulate cardiac arrhythmia at the organ scale. So the cell scale building blocks are ordinary differential equation systems. And so the schematic on the left is from Tom O'Hara's 2011 paper, which is sort of the emerging gold standard for computational modeling of the human ventricular action potential. And you can see the different ion channel populations. I hope you can see my cursor, but if you can't, I think you can probably still follow along. Different ion channel populations in the sarcolemma are represented by sort of lumped populations, fast sodium channel here, transient outward potassium channel, delayed rectifier, potassium channels, inward rectifiers, and then there are even subspaces to represent, for example, the dyad between the T-tubulus and the sarcoplasmic reticulum with resident ion channels like L-type calcium channel, and the J-rel here, which represents ryanodine receptors, which open to release calcium from the SR. So those schematics can be converted into ordinary differential equations, and essentially the kinetics of each ion channel are coupled by the membrane potential and have individual state variables and governing parameters that can otherwise be treated as independent with respect to the system as a whole. And so here's where I'll switch over to the live demo part of the presentation. So hopefully this goes off without a hitch. Veronica, stop me if nobody can see what I'm doing. So this is the underlying source code for that very same ionic model, the O'Hara-Rudy dynamic 2011 model. And this is a CARP-specific markup language, which is called Easy Markup Language, EasyML. The one thing that will jump out at you right away if you're a programmer is that it's a non-sequential markup language. And so this is the top of the source file. And there's an equation here, the governing equation for the whole system. You can see that even though we haven't defined variables like INA, INAL, ITO yet, those are still valid arguments in this syntax. And so the source file has different sections, parameters that can be modified by users. I'm going to post this all afterwards, and Veronica will send out a link, I hope, so that you can take a look for yourselves and even try and get it up and running in the simulator. There's sections for different variants of the ventricular cell models. We have endocardial, epicardial, and mid-myocardial cells. They're generic model constants related to extracellular ion concentrations and physical quantities. There are initial conditions for all the state variables. And then subsequently, each section of the file is dedicated to constants and governing equations for the individual components. So those little studded ion channel representations that you might remember from the schematic, each one of those has state variable equations and parameters. It's sort of a complex tangle of ordinary differential equation. This syntax can be converted by a program that's included in the openCARP framework into a compilable C++ source file, which I'm doing on the fly right now. Through the magic of the internet, this is all happening on my desktop machine at my office in Seattle. And so we've now compiled that C++ source into a dynamic linking object that can be coupled into the programs of CARP. And this is the first actual simulation command that we're going to run here. So bench, short for benchmark, is the single cell modeling tool, which is used to simulate action potentials in individual models. And so this command is going to run in that model, the very same model that we just compiled, that we could have changed the source code for prior to compilation. It's going to run a single beat and then plot it in the program that we use, which is XMGrace. You can see sort of the characteristic upstroke, plateau phase, and repolarization. Like I said, on the order of about 300 milliseconds is the typical APD for the human cardiomyocyte. The second experiment here will run a sequence of 5 action potentials, there's a sort of slight variation in the command line sequence here. You can see that if you look really closely, there's some subtle differences between the first and second action potential as the system settles down and moves towards steady state. But essentially, we're seeing the same action potential 5 times, which is as we expect. The third experiment here is I'm sort of trying to layer up complexity to show you how we can probe the effects of different parameters. Here, the important command line change is right in the int-par section here, where I'm saying, okay, let's imagine that there's overexpression of the late sodium channel in this cardiomyocyte, or the late sodium current, which is in fact a pathophysiological remodeling that we've studied in a previous paper that we published, and then plot those side by side with the control condition in black. We see that the introduction of this mutation has important effects in terms of prolonging the action potential duration, which could be arrhythmogenic. And then the final cell scale experiment here, just sort of give you a hint of how we sort of, string commands like this together to sort of explore the effects of complexity in different cell types. This is going to run a total of 6 simulations in endocardial, epicardial, and mid-myocardial-like cells with and without that same late sodium mutation. And then at the end, it's going to plot all 6 of them together so that we can see things like, for example, the relative effect on action potential prolongation is larger for the epicardial phenotype and the mid-myocardial phenotype compared to the red lines, which represent the, oh, I'm sorry, I said epicardial, I should have said endocardial, which is black, compared to the epicardial phenotype for the model, in which the effects of the mutation are more muted. Okay, so that was our successful foray into live demo territory. We'll return to the slides now. And so those are the cell scale components, and we can use those in order to sort of probe the sensitivity of the model system to changes in parameters, different ion channel expression levels, different drug binding effects can be simulated by modulating the expression levels of these individual ion channels, either dynamically or statically during simulations. Okay, and so computationally, what those models produce is a relatively good approximation of the behavior of cardiomyocytes studied in vitro, usually with patch clamp electrophysiology. At the tissue scale, we sort of graduate up the level up. We homogenize the system, so we have hundreds or thousands or hundreds of thousands of individual nodes in finite element models, and in each of those nodes we incorporate a copy of that same ionic model, the cell scale model that I talked about in the previous section, and so each node in a particular finite element representation of the cardiac tissue will have its own copies of those state variables, which will evolve over time in a voltage and ion concentration dependent manner. And so this system can be formulated as one or more partial differential equations, which can be solved using a finite element analysis to represent propagation of electrophysiological activity in the tissue. And the formulation that I'm showing here is the bidomain formulation that includes explicit representations of the intracellular and extracellular domains at each point in the heart. But generally speaking, we use a simplification that assumes that the extracellular domain is isopotential, and that's called the monodomain formulation. And here's an example of a video showing a propagating wavefront from the bottom of a slab of simulated cardiac tissue. These need to be solved numerically. It's very difficult to find analytical solutions because of the hundreds or thousands of state variables and parameters that come into play at the cell scale. And generally speaking, for problems of any spatial complexity, we need to use high performance computing systems in order to solve a clinically interesting number of heartbeats in these systems. Okay, so what I want to stress during the live demo part here, which I'll show momentarily, is that many different types of heterogeneity, which is sort of an essential component of arrhythmia, can be represented at the tissue scale. Specifically, the anisotropy of cardiac tissue, which is best described as sort of preferential conduction along the direction of cardiac fibers. So cardiac tissue is laminar and has well-defined striations. If you imagine the heart as a piece of meat, the sort of grain of the meat, as if you think about cutting through a steak, that's the prevailing orientation of fibers, of myofibers in the myocardial tissue. And that has extraordinary consequences in terms of the electrophysiological activity. Impulses propagate much faster along the cardiac fibers as opposed to transverse and normal to the sheet in those in cardiac tissue. And additionally, spatial variability in cell scale parameters can be something that arises as a consequence of disease-related remodeling or genetic mutations that can be heterogeneous. And the best way to think of this is that different nodes in the model can have different ion channel profiles, which can be encoded in the cell scale models. Okay, so let's jump back over into the land of the live demo. I hope I'm doing okay for time. I think I'm all right. These are the commands. I'm going to just, rather than go through step by step and show you the running of each of these, because they're a little bit more heavyweight, they require a few minutes per simulation. In this context, I'm going to show you the model that was generated using built-in tools in CARP. So this is a little bit more complicated than most people could breathe in, especially if you're on the West Coast and it's still 10 to 10. But you can see the sort of the outline of the finite elements, and you can see that what I've done here is imposed a vector field. So the fiber orientations in this model go from negative 60 degrees on the what we'll call the sort of epicardial face to positive 60 degrees on the endocardial face. And so that's a 120-degree twist, which those of you who are well acquainted with the cardiac literature will recognize as sort of the approximate profile of fiber orientation rotation in the ventricles. And so let's look at the consequences of stimulation here. I'm not going to walk through step by step, but I'm just going to show you the consequences of those different types of heterogeneity that are incorporated in the model. So on the left, we have an isotropic case where there's no effective fiber orientation. And when we stimulate the model, just walking through here with my keyboard, you see that there's a relatively concentric profile of the activation wavefront propagating through the block of cardiac tissue. And in contrast, on the right-hand side, where we have incorporated and we've included the effect of anisotropy, we see that there's this slowing of the conduction in the direction transverse to the fiber orientations. And in fact, if we flip the block around, we know that there's going to be complex interaction between the fiber field and the propagating wavefront here. And you see that that effect has been smeared out quite a bit by the electrotonic coupling between layers, so that we don't get such a prominent ellipsoid on the endocardial surface by the time the wavefront arrives here. Okay. Lastly, what I want to show you is the effect of ionic heterogeneity. And I don't have a tremendous amount of time here, but what I want to emphasize is that different regions, the software permits for different regions to have different ionic properties. And in this case, what I've done is simulated a region, a sort of spherical region on the right-hand side, starting at the right face of this block. And in that region, I've completely knocked out the sodium channel expression, which would, of course, have severe arrhythmogenic consequences if it were to happen in a patient or even induced pharmacologically in an experimental model. And you see right away the impact of the loss of sodium channel function. And you see that it also interacts in an interesting way with the heterogeneity imposed by the fiber field anisotropy, in that where the fibers on the towards the endocardial side here are oriented in such a way that the ellipsoid is stretched out towards this corner of the block, we see that it has interesting consequences. And there's still depolarization propagating here, and that would be as a result of calcium-driven action potentials. Okay, so I'm going to return to the slides here, and I'll just wrap up and give you a very brief sense of how we use these computational models. So at the organ scale, I'm not going to show a demo of this because it would put everyone to sleep watching me segment images on a Monday morning here. But we essentially at the organ scale, we want to reconstruct cardiac geometry and the distribution of diseased cardiac tissue from clinical imaging modalities like contrast-enhanced MRIs. The top row here shows a canine infarct model. The bottom row shows a human atrial fibrillation-based model. In both cases, we can extract information about the spatial distribution and burden of remodeling, so scar, either imposed as part of the experimental protocol in the dog or acquired as a part of the disease process in the bottom row for the human. And we can model the regions differently at the ionic level and at the tissue level, as I've shown you in the live demo, in order to represent the overall electrophysiological profile in the patient. So we can use these for many different things that range from basic science to translational applications. We can drive mechanistic insights about how arrhythmias happen. We can develop new therapeutic approaches, including drugs and different devices. We can use these in a translational context to stratify patient risk. So once we've reconstructed a population of models, we can pinpoint which individuals might be more likely to have a future that includes arrhythmia. And finally, we can plan clinical procedures like catheter ablation, where tissue is selectively destroyed in order to eliminate the substrate for reentry, which drives arrhythmia. And that's an example that I'm not going to go through the paper at all, but I'll point you towards this paper that we published earlier this year in Nature Biomedical Engineering. This was a culmination of my work at Johns Hopkins before I moved to UW. And essentially the idea is take an MRI scan of a patient prior to their ablation, model their heart, run a bunch of simulations, and then create a patient-specific treatment plan that can be used to help the clinicians navigate to the exact areas of the heart that need to be ablated in order to eliminate arrhythmia. And excitingly, Hopkins is now kicking off a randomized clinical trial that will test this in a controlled way. And so that's the end of my talk. I hope I didn't go too catastrophically over time. I see Veronica popped up, so happy to take any questions. Thank you for the invitation.
Thank you so much for presenting, Dr. Boyle. I'll open it up to the audience first if anybody has questions on either of the two talks. If anybody does have questions, you can just unmute yourself and give them directly to our speakers.
Hi, hello, this is Arthur from Mount Sinai. Am I audible?
Yes, we can hear you, Arthur.
So one just very low level question I had for you, Patrick, and I really enjoyed your talk. It was very comprehensible to somebody who doesn't know knows very little about the heart. So in the openCARP model, why is it better to have a lot of constants in your software rather than perhaps in configuration files?
So those are default values for the parameters, but in fact, they are all user modifiable, and pretty much anything can be made user modifiable as a parameter. Generally, it's good to set up some guardrails when you're implementing an ionic model. So some parameters might derange the ionic formulation. If you decide you want to impose a huge gradient in, potassium channel or potassium ion concentration, for example. But yeah, almost all of the parameters can be made user modifiable for sure at runtime.
I see. So even though they're in the code, they're still adjustable.
That's correct. Yeah.
So I had another question about the tissue scale model. And this is something we think about because we're building models of the biochemistry, the comprehensive models of the biochemistry inside individual cells. And, you know, we haven't got close to modeling tissues yet, but except to think about it. Which is, now, is it entirely necessary to model each cell individually? Or are there some cells which are similar enough to each other that they could be, that the models could be somehow simplified or reduced?
We don't model each cell individually. I should have stressed that, especially for this community. So at the tissue scale, there's a homogenization process that's intrinsic to the finite element formulation of the monodomain equation that we're solving, or the bidomain equations in that in that scenario. We are basically boiling down, you know, what would be representative in a whole heart of billions of cells into usually a few million finite element nodes. And then the number of cells that are comprised in each individual finite element might be on the order of thousands or tens of thousands even. And that's all baked into the homogenization process. The consequence of that is that it does make it really difficult for us to incorporate and correctly recapitulate things that, you know, data that we get from experimental collaborators, where they'll say, for example, like, okay, so we see a 20% knockdown in the connexin 43 level in the presence of this particular mutation. Well, we don't have an explicit way of changing any parameter, any single parameter in that model. And so we need to resort to saying, like, okay, well, what effect did that have on the conduction velocity? And then we can recalibrate the tensor value, which controls the sort of macroscopic property of conduction velocity. There are groups, I don't want to ramble too long here, I could literally go on forever. There are groups, there's this particularly a group at Duke by Craig Enriquez that have looked at the feasibility of, you know, tissue scale and organ scale models that incorporate like an exact, like one, you know, even intracellular space, and explicitly model the boundaries between cells. But at scale, that becomes extremely complicated and computationally burdensome.
Thank you.
So we have a question in the audience in the group chat from Taylor Pullinger. That is a question to Miguel. That is about if RIRR, or cross-induced ROS release, occur in mitochondrial networks in other tissues other than the cardiac tissue. He is also asking if the homogenization in the finite element model balance with modeling heterogeneity across cells in the tissue. So you can check these questions in the group chat. You can open it below if you need to read it, Miguel.
I have unmuted myself. So are you listening to me?
Yeah.
Yeah. So the first question is if ROS-induced ROS release has been applied to other cells than the heart. That is the question.
Yeah, I think so. It could be like that. Yeah, he's asking if the mitochondrial networks that produces the release of the ROS-induced ROS release occurs in other tissues.
Well, you know, the ROS-induced ROS release has been shown and discovered somehow in the heart, but because the organization of the network, the topology as a regular lattice is important for that. In other cells like neurons, for example, in which the networks are more reticular rather than regular lattices, there is evidence that ROS-induced ROS release can also happen, but it is not as well demonstrated as in the heart. So the second question, I don't know if it is addressed to me or to Patrick.
I think it's for Patrick, yeah.
Yeah.
Yeah, That's a very astute question, Taylor. And there's no simple answer. Effectively, what the homogenization process does is if you assign the properties to discrete regions in a model, so in the example that I showed, basically the left half of the block had control conditions, and the right half of the block had blocked sodium current, so reduced excitability. The homogenization process ensures that there's an intermediate region where you're effectively getting, an intermingling of both regions. And so and then that's determined by the electrotonic coupling, which is baked into the homogenization part of the model. And so even though you have two discrete regions, mutation and no mutation, you get a third, or sort of a spectrum of intermediate regions, and the spatial extent of that region is determined by the space constant of the cardiac tissue, which is it's not an explicit parameter, it's something that is an emergent parameter of the strength of intercellular coupling and the intrinsic excitability of the cells. But it's on the order of less than a millimeter usually. So if you have, for example, an ischemic region at the core surrounding scar in an image-based model, it's not just going to be limited to the elements where you impose the ionic changes in the ischemic region. There's going to be sort of a halo region surrounding it into the healthy tissue where there's going to be a penetrating effect. I hope that makes sense.
All right, thank you both for your incredible talks. I think we do have to wrap up in interest of time, but thank you to everyone who has attended. I hope you got a lot out of this session. Please join us again next month for our session on the microbiome. Thank you, everyone.
Thanks for organizing, Veronica.
Thanks.
Thank you.